excited to have special guests from NCBI, Genbank, and SRA discussing successful sequence submission. So we also have a special hour-long format today. We're very excited about it, especially as keepers of data. Having good high-quality sequences coming in is helpful for everyone. So previous Tools for Tomorrow webinars are recorded and they can be accessed on the BRC Consortium YouTube channel, which I'll put the link in the chat. And then next month, we'll hear from the team at BRC Analytics about differential gene expression. So I'll hand it over to John and Linda from NCBI to go into this today.
All right, thank you very much. Can you hear me okay?
Yep, sounds great.
Excellent. All right, well, hello, good morning. Thanks for having us. My name is John Tro, and I am a curator at the Sequence Read Archive at NCBI. And I'm going to kick things off today. So just by way of a little bit of roadmap, I'm going to give a little bit of background on NCBI data archives, and then I'm going to talk about biosample and bioproject records and how they can be linked to sequence records. And then I'm going to finish up with some best practices and tips for submitting to SRA. And then I'm going to hand things off to Linda, who's going to give more information about submitting to Genbank. So with that, let's get started. NCBI has several different types of sequence submissions that we accept. At SRA, where I work, we take raw reads, and those in many ways function as the base of the archive stack. And those reads can then be assembled into higher order constructs and can have other sort of annotation and functional information overlaid. And NCBI has archives for each of these data types because we understand that a sequence in isolation lacks context. And this means that it cannot be easily interpreted and is unlikely to be particularly informative. But in contrast, having a comprehensive collection of sequences allows for interpretation. And when we have multiple sequences, we can compare them to one another, for instance, using a sequence search similarity tool like BLAST. And from those alignment comparisons, we can then annotate similarities and differences, which can then prompt research into those areas. And the subsequent publication of that research puts the knowledge into the public domain, and those publications then serve as a resource to inform future research by others. And at NCBI, we try to support each of these steps in the process, all the way through to publications in PubMed that can be linked back to the specific data records. Everyone has a specific purpose in mind when they're sequencing for their project, but in many cases, the most interesting thing to do with your sequences will be thought of by someone else. And at NCBI, we want to facilitate that by making sure that each sequence has that important context. NCBI provides, in pursuit of this, NCBI provides comprehensive community sequence repositories. So we have a collection of submitted DNA and RNA sequences, as well as the matching metadata describing those sequence samples. We store both open access public data that anyone can access freely at any time, as well as controlled access human data that's available only to authorized users. And our data is submitted by tens of thousands of individual data generators and research organizations annually. So it's a lot of sequence data, providing a lot of context. In addition, we want to make sure that the data is usable, and so NCBI supports fair data practices. This means that the data are findable, accessible, interoperable, and reusable. And in support of this, for instance, we have metadata packages that are available to support community standards. So some of our biosample packages have been compiled in conjunction with other organizations and community groups to make sure that they have the relevant metadata fields for those types of samples. Our data are searchable and retrievable through NCBI and NLM resources and tools with no usage restrictions, available to anyone in the world. And our submitted sequence data are transformed into interoperable formats to make it easy to use them together regardless of how they were originally generated. So broadly speaking, what makes a good sequence record? One thing is a logical organization. This makes the records easier to use and understand, and this gets back to that idea of context. If you just have some files that are vaguely named sitting in a directory, that's not actually that useful. But if they have metadata associated with them that tells you what the content is, shows how they are related to each other, and perhaps how they are related to other records, then they become much more useful. In addition, a good sequence record has robust metadata using standard fields, formatting, and controlled vocabulary where that's appropriate. This allows for better search and filtering, and also allows for use of that data in combination with other data sets, particularly when you're working at scale. If different users are using different date formats or using different organism name formatting approaches, or if they just use the same field to mean different things, it's really difficult to make sense of them and to use those records together. And so standardizing that just makes the data much more usable overall. In addition, a good sequence record has identifiers that match those in any associated publication, but the sequence records are also fully described in and of themselves, so they can be utilized without that publication. This ensures that readers of your paper can easily replicate your work, which is often one of the main reasons you want to submit, but it also facilitates efficient reuse of that data by others to answer different questions. And if they are trying to use dozens or hundreds or thousands of data sets together, they may not have time to read every associated publication, but if the metadata is complete and standardized, they can still make sense of those data and use them together. And finally, we want sequence files that are submitted in a standard format, and in the case of SRA, with minimal editing of the sequence identifiers. And this maximizes compatibility with downstream pipelines and helps ensure that we can normalize the data, and that makes it that so that when you download it, you don't have to convert it to all these different formats into a format that's workable. That's already been done for you. We'll be talking largely today about Genbank and SRA, and Genbank and SRA contain sequence data. Specifically, Genbank is an archive of assembled and annotated sequences, and SRA archives the original read data generated by high-throughput sequencing technologies like Illumina or Oxford Nanopore. But importantly, SRA data has no annotation or assembly associated with it. So if you have reads to submit, that comes to SRA. If you have assembled data, that goes to Genbank. And both SRA and Genbank are members of the INSDC. This is the International Nucleotide Sequence Database Consortium. And so we have shared data and metadata standards with our partners in Japan and Europe. And in addition, we all mirror each other's sequences to ensure that there are copies at archives on different continents and really ensuring that full context for every sequence. So Genbank and SRA contain data records, but they can be linked to metadata records in Biosample and Bioproject. And this can sometimes be a point of confusion for submitters because Biosample and Bioproject do not themselves contain sequence data. They just contain metadata, but they can be linked to sequence data records. So a bioproject is a collection of metadata related to a single study, generally originating from a single organization or a consortium. And it just contains a description of the general study and the initiative overall. It has information about the goals, the participants of that study, and any associated publications and grants. And this kind of acts as a high-level accession that collects links to data in different databases so that you can find all of it in one place. So sort of a one-stop shop accession. Whereas a biosample is, again, just a metadata record. It does not itself contain sequence data. And it's a description of a distinct biological sample. It's the source material you used when you were doing your sequencing. And so this contains metadata such as the organism name, the collection date, the location where it was collected, etc. And having the metadata exist in these separate objects, biosample and bioproject, has a few advantages. It allows us to capture the metadata in a structured way by promoting the use of controlled vocabularies and sample attribute field names and values. So regardless of whether you're submitting to SRA or Genbank or somewhere else, the metadata can be captured in a standard way. It also allows you to link the metadata to corresponding experimental sequence data across different archival databases to support cross-database searches. So if you have linked your SRA records and your Genbank records to one bioproject, by searching that project, you can find links to both of those data sets and understand that they are related. And it also can reduce submitter work by enabling one-time upload of these description and metadata details. And then you just reference that sample or that project when appropriate when making deposits to the data archives instead of having to re-enter it each time. So to look at this in a little bit more concrete way, these are all separate records in separate databases that can be linked up. And the bioproject and the biosample records on top are metadata records, whereas Genbank and SRA contain the actual sequence records. And so, for instance, in SRA, it's required that each run be linked to exactly one biosample and one bioproject. You can't submit without linking to these records. And in addition, if you are submitting both to SRA and to Genbank, you can link your records to the same sample to indicate that they were generated from the same biological source material, and the same project to indicate that they are part of the same overall study or initiative. One thing to note here is that not all Genbank records have an associated bioproject and sample, but they can be linked to those. And if you are submitting to multiple databases at NCBI, that's highly recommended to make your data maximally useful. And for an example here, this is just an example of what a search might look like when searching at the NCBI's main page. You can search for a bioproject or a biosample accession on this page. You can search for any other accessions as well, like a specific Genbank accession or a run accession. But here we've searched a bioproject, and you can see that you get hits to the different databases. So we have a link in PubMed Central to a publication. There's assembly data, the bioproject itself, there's genome data, there are SRA records. And so you can quickly see all of the related data. And if you were to click on the top link here, that would take you to the bioproject page itself, which gives you the sort of overall description and grant information, but will also contain links to these different data types as well. So you can easily find all the related information. So drilling down a little bit more into the SRA data model itself, I told you that every record is linked to a biosample and a bioproject. If you've ever submitted to us, you've seen that the biosample accession format is they all start with SAMN and then a number, and bioproject is all PRJNA and then a number. And these link to SRA records. The run is kind of the atomic unit of SRA. It's essentially a container for sequence files. And so that's the actual accession you would feed to like our toolkit, for instance, if you were going to access the data in FASTQ or SAM format. But there's this other object called the experiment. And when you're submitting, you generally don't have to worry about this at all. We kind of take care of creating these on our end. And the vast majority of SRA submissions, there's sort of one biosample linked to one experiment, linked to one run. And when you're submitting to us, most submitters are submitting in the web interface. So you're sending us a spreadsheet with your metadata. That's just one row. That sample experiment run is one row on that metadata sheet. There are some circumstances where you might want to have multiple experiments linked to the same biosample. So for instance, if you have one sample that was sequenced on two different platforms, you have Illumina reads and you have Oxford Nanopore reads, that would be a case where your spreadsheet would have two rows. And both rows would have the same sample listed on them, but you would have distinct sequencing information and file information on each row. I'll just mention here that we do also offer submission using API programmatic submission of XML. If you are submitting large amounts of data over longer periods of time, it's generally worthwhile to set that up. It allows you to submit programmatically. You don't actually have to come to the web interface to submit. It's much better for high volume submitters, and we're happy to help you set that up. But having seen what the basic structure of an SRA submission is, what should you prepare prior to submitting? First and foremost, a project name and a description. Or have an existing bioproject accession. If you've submitted to this project before and you're here to add more data, there's a question at the very beginning of the SRA submission asking if you've already have a project. You answer yes, you enter the accession, and then you don't have to enter any more information about the project. But if you haven't done that, you'll enter your name and description of the project, and then optionally, you can enter other information like participants and grants, although certainly you can add that information later as well. You'll also need to choose the type or package for your samples, or if you already have biosample accessions, you'll skip this step and you'll just enter your existing accessions. But if you haven't submitted for these samples before, you'll need to pick a package. And the package is really just a set of recommended and required attributes that are most relevant to that type of sample. So for instance, if you have a viral human pathogen, you would certainly submit using the human pathogen package, or if you have a virus that's not a human pathogen, there's just a more generic viral package. And most of the time, it will be fairly clear which one you should pick, but you can go ahead of time and preview the packages and the definitions for all the attributes here at the page listed, and I'll talk a little bit more about this in a future slide. You'll also need to gather the sample metadata for those biosamples and make sure that it's unique for each biological sample. And importantly, this uniqueness can't come solely from the sample name. We don't want you to encode important distinctive information just in the name. It's fine to have it there, but we'd like you to also encode that in the other attributes on the sample because those are best for index search and filtering later on. Then you'll gather your sequence metadata. So this is which sample each of your files should be linked to, information about your sequencing and library construction protocol, library names, sequencing platform, etc. And then finally gathering your files in an accepted format. FASTQ, BAM, and CRAM are the most common, although we do accept other file formats such as PacBio H5. One note here is that barcoded data should generally be demultiplexed prior to submission so that there's one or more unique files for each biosample. This helps ensure that users can adequately match up each of your sequence records with specific sample information. A little bit more about choosing an appropriate biosample package. As I mentioned, what we call a package is really just a set of attributes that are related to that type of sample, because you would want different attributes if you're submitting a model organism than if you're submitting some kind of metagenome or a human pathogen. They sort of guide you in providing appropriate attributes to describe your samples. When you're choosing this, there's a description of each, and generally you can just choose the package that seems most appropriate for your samples. If you're not sure when you're submitting, there's a search bar on that page, and you can enter your organism name, and it will actually filter the list of packages to those that are most relevant to your sample type. One thing I do want to note here is that there are specific packages for metagenomes. So if you're submitting any kind of microbiome or metatranscriptome, 16S, ITS, all of those types of things should generally be submitted as metagenome samples. And again, you can preview the different packages and the definitions of those attributes at this website. So when submitting to biosample and bioproject, submitters are responsible for a few things. They need to provide complete metadata using the accepted formatting, and that we help with that. There are some validations on the page that help make sure you're getting that correct. You need to ensure there's no personally identifiable information in the metadata. This is particularly important for, say, clinical samples. You don't want to have any names or government issued ID numbers or any kind of patient IDs unless they've been appropriately blinded. And then you would need to use NCBI services to submit that metadata. You can specify a desired future public release date. You don't have to release it immediately. You can ask us to release it in a year, for instance, and you can always change that. You can come back and say, oh, I thought it was a year from now, but really my publication came out early. Can I release it now? Or if your publication is going to come out later, you can ask us to push that release date out. You can change it. When it comes to processing your samples on our end, we're going to perform both automated and manual checks of the metadata to ensure that the integrity of it and the overall quality of it. Once it's passed those standards, we will assign accessions to those records. And then we will hold those records in a private status during processing and prior to public release. We might halt processing of your submitted metadata records for various reasons. One is that if we find unrecoverable errors in the metadata formatting or values while we're checking it, it might go into an error state that will halt it from processing. And in those cases, we may contact you to ask for clarification or to make updates to it. Or we may also halt processing if the submission is found to duplicate existing records. We don't want the same records, either sequence or metadata, being submitted multiple times. The hope is that each accession will be specific and unique. So if we find duplicates, we'll generally flag that as an error and ask you to address that. When submitting to SRA, some general best practices are, first of all, I mentioned this before, but I'll say it again, that SRA does not accept assembled or consensus data or contigs. Assembled data should be submitted to Genbank, as you'll be hearing shortly. But if you have the reads, those should come to SRA. And they can be the raw reads, or they can be somewhat processed as well. They don't have to be absolutely raw. You should make sure that just as with your metadata files, the files and file names should not contain any personally identifiable information. This can cause halting of processing of your submission and force you to resubmit new files. Files can be compressed using gzip or bzip2, but we ask that all of the compressed FASTQ files be less than 100 gigabytes in size. If they're larger than that, we generally ask that you split them prior to submission. And if you need help understanding how to submit those, we can assist with that. In addition, if your study that you're submitting data for has more than 5 terabytes of data, we ask that you split that across multiple submissions instead of submitting it all in one giant submission. This is largely because the area where you'll be uploading your data is as kind of a temporary staging area. It doesn't stay there forever, and it's a shared area. Other users are going to be uploading data in that same general area. And so if you upload 60 terabytes of data and there are some problems with your files that make them take longer to process than normal, and that data is sitting there the whole time, it can make it difficult for everyone to have enough space. So you can submit your data across multiple submissions, and they can all be linked to the same bioproject as I mentioned before. So if you submit that way, when users are actually searching for your data in NCBI search, it will be indistinguishable from having just submitted all of them in one submission. You should plan to use your primary email address in your communications with us when you're in submission portal. There you have a login account there. Make sure the primary email address is one you actually check. That's where we're going to send updates about your submission, your information about your accessions, and if we need to talk to you, that's who we're going to email. So please make sure you check it. There's a secondary email field there as well. We recommend that you have an email address there that is different from the primary. This just makes it easier to make sure you can always recover your account if, for instance, your primary email address is associated with your educational institution and you graduate and leave and you no longer have access. Having the secondary always gives you another way in. Please make sure that every organism or metagenome, time point, tissue type, treatment type, that each of these things have their own sample. So if you have the same organism at 3 different time points, each of those should be its own biosample. And then finally, if you already have a project for your future publication, please don't create a separate project specifically for SRA. If you've submitted to Genbank already and you have a genome submission there and it's got a bioproject, when you submit those reads to SRA, use that same project. And likewise, if you submitted to SRA first and then you submit to genomes, make sure you use the same project. This will ensure that all of your data are linked up appropriately. So when submitting to SRA, again, submitters need to submit intact files in one of our accepted formats, and you should ensure you have the authority to submit that sequence data. Don't submit someone else's data without their say-so. Make sure that you have the right to do that. And you can use our services to submit sequence data and metadata. We accept uploads via direct cloud upload, via FTP. If the files are small, you can upload directly in the web interface. So there are several different methods you can use to get your data files to us. Just like with bioproject and biosample, you can specify a future release date that you would like. You can come back and change that release date, and you can even change it yourself. So if you say you want to release it in a year when you first submit it, and later you decide it needs to be released in 2 months, or you decide it needs to stay on hold even longer, you can come back and make that change on your own. When we're processing your data, we're going to do automated and manual checks on your data in some cases to ensure data integrity and overall quality. Once it passes those checks, we will assign the accessions and send those to you for your records, and then we'll hold the sequence data in a private status during processing and then prior to your desired public release date. There are a few reasons we might halt processing of your submission. So if you requested it, for instance, if after submission and before the data have been released, you decide there was an issue or you want us to discontinue those records, we can assist with that. If we find unrecoverable errors in the data formatting, if they are truncated, for instance, or there are formatting issues, your submission will go into error, and we'll contact you to find out how we best need to approach correcting that. And finally, if the data are determined to duplicate existing records, we will also halt processing of your submission. Because again, we don't want to store the same sets of reads with multiple accessions. And then finally, I want to talk about a couple of SRA features that I think not everyone is familiar with, but that can be pretty useful for some submitters. One is the human contamination screen. You can contact us and ask us to activate this for your bio project. And we will, when we load your data, we will scan all of the reads and then we'll redact any human reads that are in the sequence. We highly recommend this for clinical samples or human metagenomic samples because these types of data almost always have some contaminating human reads. It's activated on a per bioproject basis, so any past data you've submitted to SRA for that project or any future data you submit to SRA for that project will all be screened and the human reads will be redacted. And I've got a footnote here. Software is actually public, so if you want, you can download it and run it yourself on your end on your FASTQ files if you prefer to scrub before submitting. In addition, in the submission portal, we have shared submission groups. And these are through submission portal, so it's not just for SRA. It works for submitting to biosample, bioproject, et cetera. But it allows you can contact us, ask us to make a group, or you can make a group on your own. And this allows a group of users with distinct logins to all collaborate on one or more submissions. And this can be really useful for lab groups or consortiums who need to work on submissions together. It allows you to have your individual accounts. You don't need to share login information or passwords, but you can all work together on these. And you can kind of manage the group over the life cycle. So if, for instance, you're managing a lab group, you could have the admin of the group be the PI, and maybe there would be someone else, a lab manager who's an admin, and they can invite new members to the group as needed when new members come to the lab. If members leave the lab, they can remove them from the group and they can change people's permissions, and just allows you to manage your submissions and your data over their life cycle along with other people. And then finally, we have the sequence taxonomic analysis tool, or STAT as we usually call it. And this is a K-MER-based tool that we use to assess the taxonomic diversity in submitted SRA data. And this is done independent of the submitted metadata. So every data set that is sent to us has a biosample. It's been declared to be some organism, but we're actually looking at the reads individually using these K-MERs to determine what it looks like the reads are actually from. And this can be a really interesting tool to look at the content of your own data as well as the content of other users' data. We conduct this analysis on all submitted SRA data that's converted to our normalized format. We present these results on NCBI web pages, so you can see this information in the SRA Run browser. Before your data are released, you can even see the top 10 hits from this for your own data in Submission Portal. So this can be useful to get a quick window into what it looks like is in your data before you release it. And we have the full set of results available in the cloud, and these are searchable using either Athena in AWS or using BigQuery in Google Cloud Services. This allows you to search for hits to your favorite organism of interest across all of SRA, all of our samples. And so you can either just sort of search in a raw way for runs that had hits, or you can limit it and also search via submitted metadata terms. So within certain collection dates or certain collection areas, then look and find all the runs that had hits to your specific organism of interest. And this can be a pretty interesting way of looking at what's inside the data before you download it and actually dump it. And we've got a note here at the bottom, number 2 is a link to the paper that describes this tool. But with that, I'm going to pass things off to Linda.
Thanks, John. If you could go to the next slide. My name is Linda Yankee. I'm the Genbank Curation Team Lead. I'm also a product owner for our internal processing tools for Genbank. We're going to transition now to the best practices and tools for the submission of assembled and annotated sequences, including a preview of our upcoming submission portal web interface. to replace our old Bankit tool, which you may be familiar with. I'm going to be directing this talk to mainly discuss viral sequences because of the audience, but this tool will be available for other types of sequences that Bankit used to accept as well. Next slide. So I think, as John mentioned, the key to a successful quick submission process is the preparation of the necessary files. We highly recommend submitting similar sequences together as a single submission. A lot of people don't know, if you have, say, 1,000 poliovirus spike genes, these can all be submitted in a single instance. Sequences should be in FASTA format and can use the isolate as the sequence identifier to provide ease of tracking. The sequence identifier must be the same across the FASTA, source, and annotation files. Information about the source organisms of the sequences can be provided in a table or spreadsheet format. There's many source modifiers that you can use that are available for according to INSDC documentation. But at a minimum, the source for assembled virus submissions should contain the organism name and information about where and when the virus was collected, including the date, locality, host, and/or the physical environment. Similarly, there are many features that can be added according to INSDC documentation, but the major use case is for coding regions and genes, so that will be the focus of all of my examples. Annotation is accepted as a 5-column feature table, and details on this format are available online. Or if you're using the web interface, you can use forms to add the annotation instead of preparing a feature table. Finally, you'll be asked to provide information about submitting authors and any relevant publications describing the primary sequence and analysis of the data. The format that this information is provided will be slightly different depending on which submission path you choose. Next slide. So as John mentioned earlier, including this information really is a key component of the usability of the data. In addition, adding a coding region can provide a check of the nucleotide sequence itself by detecting frame shifts or contamination or misassembly. The source information is essential for placing the data in space and time to track pathogen spread and viral mutagenesis. Incorrect data also propagates and can affect other researchers' work. For example, someone trying to design primers using incorrect sequence data will not be very successful in their experiments. Incorrect sequence data could lead to artificial divergence in a viral variant tree, or a lack of source metadata could result in an inability to track viral spread. Consistent, verified data is truly a key to scientific discovery. Next slide. So therefore, it's not surprising that issues with these can be the main reasons for a delay in accepting and processing viral submissions. We ask that all of these issues be addressed by the submitter before sending the data to avoid back and forth with submission staff and to expedite your accession number assignment. In particular, checking sequence and any annotation against public data really is essential. While we've included more directed submission dialogues, clearer help documentation, and more built-in validations into our new submission tool, prior sequence analysis is still an important step to success. Every tool that Genbank staff uses for analysis and validation is publicly available and can be used prior to submission. Next slide. Okay, so there's multiple ways that you can determine and check annotation for your submission. VADR was developed by Eric Noraki at NCBI and is available in GitHub. It will output a 5-column feature table that can be directly used in submission. VADR also compares, so VADR works by comparing input sequences to model sequences and transfers the annotation from the closest model to the input sequence. It has analysis based on both nucleotide and protein comparisons and outputs detailed alignments and alert reports. VADR can produce complex annotation required for viruses, such as coding regions with ribosomal slippage like ORF1ab and SARS-CoV-2. Also, it can add mature peptides, which can sometimes be a struggle to add independently. Since the annotation is based on set models, the output is consistent for things like product and gene names and coding regions that are present within a genome, therefore greatly increasing data reuse. And NCBI Virus is planning to release a web version of VADR soon. Next slide. So the key for all automated annotation tools is having good model sequences that incorporate natural viral variations and consistent annotation themselves. The number of VADR models has been expanded, and the additions can be followed in GitHub and are listed here. I'm not going to sit here and read out all the viral names. While using family level models like Coronaviridae is more efficient than having one model per virus, it also may produce some alerts that can be ignored due to viral diversity across the family. Similarly, viruses that mutate frequently and are highly repetitive, like Mpox, may produce alerts that can be ignored after review. The addition of models by the Gruninger Group has greatly leveraged community engagement to expand automatic annotation generation to specific highly important viruses to human health. We also can update existing models or alert parameters based on community tracking of mutations. We actually had an example of that this week. There are certain SARS-CoV-2 BA 3.2 viruses, apparently, that have a larger than anticipated nucleotide deletion. VADR was flagging these as problematic. Once we were alerted to the fact that this was an incorrect alert, we were able to quickly update VADR and our automated annotation pipeline to no longer flag these sequences as problematic. Having input from stakeholders is essential to the success of the development, maintenance, and expansion of automated pipelines. We really do want and need your input for this. Next slide. So there are alternative methods for viruses that do not yet have a VADR model. We have multiple help documents on using BLAST to determine and validate coding region annotation. Online, you can use the CDS feature checkbox as shown here to allow a quick visual of sequence changes that can dramatically affect essential proteins and should be re-examined. There's also non-web methods of using BLAST for at-scale analysis, such as Elastic BLAST in the cloud. All of this is documented online. While viruses do mutate frequently, frame shifts at the ends of sequences or that dramatically change an amino acid sequence of an essential protein, as shown in this example, to the point where the protein couldn't even fold or be made, should be re-examined and supported with evidence prior to submission. If you are submitting multiple sequences, you can determine the annotation of one sequence and propagate this annotation using a multiple sequence alignment like MAFT or other commercially available alignment tools. Alignments can also be used to detect potential frame shifts or low quality sequence at the ends of nucleotide sequences. The potential to use AI to develop more automated sequence analysis tools is really an exciting prospect and would be of great value to the scientific community at large. Next slide. Okay, so now you've collected all of the information that you'll need for your submission. You can choose a submission tool based on the type of data you have and mainly your preferences for tools. Table to ASN is a command line utility that inputs the 4 types of files from my second slide and generates an ASN1 file that can be used for submission. This includes a validation suite that will output alerts to be reviewed prior to submission. Currently, we're only supporting FTP submissions for flu and SARS-CoV-2, but most of our submissions come to us via our web-based tools. Many of you may be familiar with our submission portal wizards that provide automated feature annotation using VADR for some frequently submitted viruses of importance to surveillance and human health. Coming very soon, we're going to transition submitters from our legacy program, Bankit, which, by the way, I think is over 25 years old, to a new submission portal, Genbank, for all other viruses. This will provide several advantages to submitters, the most obvious of which is providing a single unified system where all submissions can be tracked. If you've already submitted your SRA data, you can more easily incorporate links to the bioproject and biosample and SRA in this new interface. There's also the opportunity to use shared submission groups, which John talked about earlier, to coordinate submission of different data types amongst your collaborators. We also have incorporated a lot of user feedback to improve the overall functionality of the tool and to ease submission bottlenecks. Next slide. Okay, so all screenshots that I'm going to be showing you are from our test system. So there may be slight changes to language prior to final release, but there shouldn't be any major changes. When you first arrive at the new submission portal Genbank, you'll see a very streamlined landing page. The first thing you're asked to do is to select the organism type for your submission. So for this example, of course, I selected virus. Once you make the selection, the system immediately begins tailoring the workflow to your data type. You are then asked whether your virus is one of the organisms supported by an automated pipeline. This step is important. If your virus is supported by an automated pipeline, the system will guide you through a workflow that incorporates automated annotation and targeted validation checks specific to that organism. If your sequence is from a different virus, currently you're being redirected to Bankit when you click on that any other virus. In the near future, you're going to remain entirely within Submission Portal Genbank. The reason we ask for this information up front is to eliminate unnecessary fields and guide you only through options that are biologically relevant to viruses in this example. Viruses don't have mitochondrial D loops, for example, so you'll never see an option to add a D loop in the virus workflow. This organism-specific routing allows us to present metadata prompts, annotation dialogues, and validation checks that are directly relevant to viral submissions, helping to reduce submission errors before review even begins. Other major improvements in the submission portal include the ability to reuse publication and author information from previous submissions, which can save significant time for frequent submitters. And importantly, for former Bankit users, if an issue is detected during pre-accession review by curation staff, you will now see a fix button, which allows you to reopen and correct your submission instead of starting all over from scratch. Next slide. So Submission Portal Genbank maintains a tab structure across the top of the page, which you may be familiar with from other portal interfaces. You can clearly see where you are in the submission process. Each tab corresponds to a major component of your submission, for example, submitter information, source metadata, annotation, and review. Unlike Bankit, you can navigate to earlier tabs and change data without losing your progress. This is particularly helpful if you realize you need to adjust something after reviewing later sections. Within each tab, you will either see structured forms to enter information directly or options to upload prepared files. I'm not going to go through every form in detail today, but I'd like to focus specifically on the source modifiers and annotation tabs because those are directly tied to the best practices that we discussed earlier regarding metadata completeness and coding region validation. Next slide. Okay, this slide may look a little overwhelming at first glance, but what it's actually showing are the three different methods available to provide source metadata. Importantly, you will not see all of these dialogues at once. The system presents the interface dynamically based on which methods you choose. And I tried to signify that with the little blue arrows. The method you choose really depends on how your lab stores and manages metadata. If you've already prepared a formatted source modifier table, you can upload that directly and the system will validate it automatically. If you prefer a more interactive approach, you can use an editable table within the portal. Note that required fields are clearly marked with asterisks and tooltips indicated by the question mark icon provide short guidance. For example, because host is required for viruses, if you click the tooltip, you'll see instructions that for environmental isolates, you should just enter the word environment as the host and then provide more detail in the isolation source field. For example, wastewater or sewage or whatever. You also have the ability to add additional columns to link related accessions, such as bioproject, biosample, or SRR numbers during submission, or to add other relevant source information. If you use the isolate as the sequence identifier in your FASTA file, and all the other data across all the sequences you're submitting is the same, you can use a simple form to add that same information in one form. This option is also convenient if you're only submitting one sequence. Next slide. The annotation tab follows a similar philosophy of flexibility. If you already have a 5-column feature table prepared, for example, the output from VADR, you can upload that file directly. You may also upload protein FASTA sequences to annotate coding regions automatically. For viruses that are highly annotated, such as Mpox, we strongly recommend uploading a 5-column feature table to preserve complex annotation structures. Next slide. If you do not already have a feature table, or you can annotate directly within the portal using structured forms. This also can be a more appealing option if you're submitting single gene sequences. In this example that I mocked up, there's 3 sequences, each containing a partial spike gene from poliovirus. I've chosen to annotate all sequences simultaneously and added a CDS, or coding region feature. When you click the Add Feature CDS button at the bottom, a dialog appears prompting you to define the coding region. You can either enter the specific nucleotide locations of the start and stop, or indicate that the CDS spans the entire sequence. You can also select strand orientation, indicate whether the sequence is partial at either end, and then provide the protein name and optional gene symbol. This structured approach helps prevent formatting errors that sometimes occur when annotation is prepared manually outside the system. Next slide. Regardless of how you choose to add the annotation, by either uploading the 5-column feature table or by using the forms, the system performs validation checks and then displays a feature preview. This preview resembles how your annotation would appear in the final Genbank flat file. In this example, the CDS for sequence 2345 is flagged because it contains internal stop codons. You can see the warning message displayed clearly at the top of the page, and the affected feature is also highlighted within the preview itself to draw your attention. You'll notice that stop codons appear as asterisks in the translation itself. As the tooltip explains, when you expand it, you can click on Accept at the bottom of the page to generate edit dialogues where changes can be made. Next slide. And here are the edit dialogues. The edit dialogues allow you to modify an individual annotation without affecting any others. You can also add additional annotations or delete the current annotations and start all over again to add new. It also clearly indicates any annotations that have a validation warning. By clicking Edit beside the CDS with the warning, you can change the nucleotide coordinates, adjust the reading frame, or modify strand orientation. In this example, simply changing the reading frame to 2 eliminates the internal stop codon error. Once you hit Accept at the bottom of the page, then the system will revalidate the feature automatically so you can confirm that if the issue has been resolved and there are no further warnings before proceeding. Next slide. Once you've completed your submissions, what happens next? So your data will be reviewed for accuracy, content, and format using a combination of automated and manual methods. Much more validation has been put into Submission Portal Genbank, and dialogues have been improved to assist in a smooth submission process. Again, all programs that we use for post-submission data analysis are publicly available and can be used prior to submission for speed processing. Following an initial review, accessions will be assigned. Like John's slides for SRA, Genbank may halt processing of a submission for the same cases that John listed. Next slide. Once your submission is complete, you can track its status directly within Submission Portal Genbank. This screenshot shows multiple submissions in different processing states. Although I did not assign unique titles in my testing examples, you have the option to do so, which can be extremely helpful when managing multiple submissions simultaneously. You can also view your SRA, biosample, bioproject, genome, and Genbank submissions together in a unified dashboard once this is released. Notice the presence of the Fix button next to a submission that has not yet been assigned an accession. If you click this button, you can reopen the submission, upload corrected files, modify source data, or update annotations without restarting the process. Once accessions are assigned, they appear in the dashboard along with a downloadable file mapping sequence identifiers to accession numbers. When processing is complete, the status changes from processing to processed, giving you clear visibility into the life cycle of your data. Next slide. NCBI's mission is to accelerate scientific discovery and advance health through data-driven research. We do this by providing an integrated ecosystem of tools that support researchers at every stage, from sequence analysis and validation to data submission and processing, to powerful retrieval and downstream analysis resources. High-quality databases begin with high-quality submissions. The work you do before and during submission directly impacts the usability, reproducibility, and long-term value of the data for the global scientific community. Today, we've focused on best practices for SRA and assembled viral submissions, but these are just one part of the much broader NCBI infrastructure. We also support bacterial genome submission and processing, pathogen resources, specialized platforms such as NCBI virus and our pathogen detection resources, all designed to make data more discoverable, comparable, and reusable. NCBI serves as an essential scientific infrastructure. The data you submit today becomes the foundation for future surveillance, diagnostics, evolutionary studies, and public health responses. If there are specific tools or resources you would like to learn more about, we really do welcome your feedback. Knowing what is most useful to you, to your work, helps us prioritize future training and outreach efforts. And then next, John, we'll just finish up with the acknowledgement slide. There's really way too many people involved in this work to list everybody individually. So in some cases, only groups are listed. But thank you very much for your time and for providing any feedback.
That was awesome. I know the chat has been very busy with questions, but we have a few minutes. If anyone has any other questions they'd like to ask, everyone should be able to unmute and ask them if needed. But thank you so much. Nancy Wilson just asked, it sounds like you prefer that we dehost human reads prior to SRA submission. Does it matter how we do this, or do you prefer HCS?
That's a great question. It's not preferred necessarily. You certainly can if you would like. We provide the HCS tool if you want to use that. If you have other methods that you prefer to use, that's fine. But it's not necessarily a preference. We're happy to activate the HCS on our end. So it's really, I would say it's up to you what your preference is.
Vince, can you provide more information to answer Curtis's question regarding VADR in the models?
The HSV1 model is only against HSV1. If you have human ORFs inserted, it will not be able to recognize those, and it'll probably report them as an error and fail them. So no, it cannot pull from more than one reference at a time. And there are no human references in the VADR model database.
And I see Liliana asked for discussing bacterial genome annotation and eukaryotic pathogen genome submissions and annotations. I assume that you guys would like a kind of an overall on the submission process and the use of PGAP and EGAP, which we can definitely contact people to put you in contact with people to prepare that in a future talk if you would like.
Yeah, Linda, thanks. It's Liliana. That would be great, because I know our audience is broader than just viral. So we have a lot of people that would be particularly interested in bacterial and eukaryotic pathogens, as well as any metagenomic data submissions and plans for annotations there, because a lot of what we do is host pathogen data sets. So knowing anything from metagenomic and all of the clearing of reads, et cetera, would be really useful.
Well, if no one has any more questions, we can. Oh, Curtis, I know that you're willing to speak up. You can just ask the questions out.
Yes, can you hear me okay?
Yes.
Okay, great. Yeah, thank you. I joined a little bit late, but caught the last half of this presentation. Thank you so much. I'm just curious if the web-based submission portal will be able to handle flu samples where not all segments are present.
So
there's sometimes where that happens where it doesn't get sequenced. So
the automatic flu annotation pipeline that is currently available will accept segments where not all 8 segments are missing. The one caveat to that is that if you have not sequenced either the hemagglutinin or the neuraminidase, segment 4 or segment 6, you don't have a serotype. So the automatic validations are right now requiring a serotype. The workaround for now is to put in missing, and then we'll fix it later. If you were to submit this data using the new web interface, you would have to provide annotations because it is for generic viruses and doesn't have the CDS automated annotation.
okay, great, thank you. And I assume it would accept the VADR annotation files for flu. Okay.
Yes. I mean, in general, we prefer greatly that you use the automated pipeline, but if there is these corner cases like not having segment 4 or segment 6 or certain reassortants, they'll have to be done with the generic virus wizard instead.
Okay, good to know. Thank you so much.